Hello, in this video I will present you a proposed approach to analyze pigmented tumors with dermoscopy. So it will be a somehow introductory presentation on the application of dermoscopy, the main application of dermoscopy, which is for the assessment of pigmented tumors. Before starting, I would like to share with you my way of thinking to a very basic question, which is if the dermatoscope is a magnifier. Of course it magnifies, but is the dermatoscope a simple magnifier? Well, I would say no way, because this is what a simple magnifier does. So this is the clinical morphology, naked eye of skin tumor. This is a magnifier that magnifies what you already can see with your naked eye, just becomes larger. Okay, that's the magnifier, and this is no way what dermoscopy does. Dermoscopy does not magnify what you see with your naked eye. Dermoscopy brings to your eye structures, features, colors that were completely invisible to your naked eye, and that's why dermoscopy requires a specific training, because all these structures, all these features are not well known from the macroscopic morphologic aspect, and of course they have their own significance. So these structures that we see dermatoscopically practically consist of a shape and a color. For instance, here in this image here we can see some roundish structures, some linear structures, and we can also see red color, blue color, brown color, white color, and so on. So structures and colors. Of course, both are very important, both structures and colors, but especially colors are very, very informative in dermoscopy. And these colors that we see dermatoscopically arise from skin chromophores. So we see colors because in the skin there are some substances that have a chromatic effect, a chromatic result, and the main skin substances that are chromophores are these three: melanin, of course melanin, but also hemoglobin, blood, and keratin. There are additional substances with a color effect, but these are the three main ones. Melanin, of course, is the most important, and we know that the color corresponding to melanin is mainly brown, obviously. That's true, especially when the melanin is located more or less at the level of the dermoepidermal junction. When it's located more superficially, the result is black color. When it's located deep in the dermis, the result is a blue color. Hemoglobin, in contrast, usually results to red color, and occasionally, if it's located deep in the dermis, it might be more purple or bluish. And if it's very superficially, or if there is thrombosis of the blood, then the color might be black. And finally, keratin. The color of keratin is white and yellow. It becomes yellow when it's admixed with serum. Otherwise, the color of keratin is white. So these are the main colors that we see dermatoscopically, and this is the correspondence in terms of chromophores. In this presentation, we will focus on pigmented tumors, so on tumors that usually display melanin or contain melanin, and the colors we speak about are these ones: black, brown, darker light, gray, and blue. I would just like to remind that melanin, the presence of melanin, does not mean that the lesion is melanocytic, because melanin, of course, is produced in melanocytes, but then it does not remain only in melanocytes, it goes also in keratinocytes. So melanin does not necessarily mean a melanocytic tumor. Now, let's start with a few basic structures, pigmented structures, starting from black color. As I said, black color usually corresponds to something very, very superficial. And as already mentioned, when I was speaking about the chromophores, black might result from superficial melanin, or black might result from blood, such as this hemorrhage. You see these blood spots of black color. Or the next one, which is a thrombosed angiokeratoma, in which the lacunas became black in color, so it's blood again. So in both of these occasions, of course, histopathologically there is blood very superficially. That's why we see these black globules. So this is the first criterion we see, and here we see a different version of the same criterion. Black globules again. Look at the periphery. Globule, 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 black globule. Okay. Of course, in this scenario, this does not correspond to blood. This corresponds to superficial nests of melanocytes. So this is the first criterion we see. Black globules. Histopathologic correspondence is either blood or superficial melanocytic nests, tumor diagnostic significance, angiomas, vascular lesions, and Spitz reed nevi. Next structure, we still remain in the black color. Here in the center we can see a black structureless area, or if you prefer, black blotch. Okay, so what should this correspond to? This should correspond to large amounts, large concentrations of melanin. That's why we have a large black area. Where? In the epidermis. Okay, this should be the histopathologic correspondence, and this is the histopathologic correspondence in black nevi that we see in dark skin type individuals, or in Spitz nevi, the one we saw before. Of course, there are black globules at the periphery, but there is also black blotch or black structureless in the center. Okay, so black color can be seen in black nevi and Spitz nevi. Here is the correspondence: large amounts of melanin, very, very superficially located. And of course, black blotch, black structureless, can also be seen in melanoma, obviously, again corresponding to the presence of melanin in the epidermis. This is a biologic phenomenon that typifies melanoma, the pagetoid spread, the upward migration of melanocytes in the epidermis. But of course, in melanoma, the difference is that the blotch is not symmetric, centrally located as it was before, because obviously the pagetoid spread is a malignant phenomenon that does not follow any kind of rule. It occurs without any sequence. It occurs randomly. That's why the blotch is eccentrically located, or we see multiple blotches, for instance, in melanoma. So the difference between a regular blotch and an irregular blotch. The second criterion we learned was black blotch corresponding to pigmented parakeratosis, to the presence of pigment in the epidermis, and diagnostically it corresponds to regular nevi, Spitz or reed nevi, and of course melanoma. Let's move to brown color. Brown color almost always corresponds to the presence of melanin at the level of the dermo-epidermal junction. And let's see here a few results, starting first from the diagnosis. So here you can see a solar lentigo. In the next image you can see a melanocytic nevus. In the next image you can see a dermatofibroma. So in all these three tumors there is something in common morphologically, and what is in common is the presence of brown network. Why do we see precisely the same feature in three different tumors? Because histopathologically in all of them there is a continuous presence of melanin along the dermoepidermal junction, together with a normal anatomy of the junction, a normal papillomatosis of the epidermis, with the reed ridges and the dermal papillae, reed ridge, dermal papillae, okay, and continuous presence of pigment along the dermo-epidermal junction. This is the histopathologic correspondence of brown network. Of course, we see brown network also in melanoma, and in melanoma this network is atypical. And now if you consider why this is atypical, it is explained by what I showed you before. In order to have a typical network, as I said before, we need continuous presence of melanin plus normal papillomatosis. In melanoma, the papillomatosis of the epidermis is not normal because, for instance, two neighboring root ridges might be united, might collide one to the other. This is the so-called bridging, and therefore the whole of the network here will be lost. So that's why in melanoma we see atypical network and not a regular homogeneous network all over the surface of the lesion. So brown network corresponds to pigment in the basal layer plus normal papillomatosis, can be seen in nevus, dermatofibroma, solar lendigo, and of course melanoma. Next criterion, brown color again, globules, brown globules. They are roundish structures, therefore they should correspond to something roundish histopathologically, and that's true. They correspond to nests of melanocytes, quite large nests of melanocytes, located where? Located almost at the junction, immediately below the junction. That's why the color is brown. Otherwise the color would be blue, okay? But it's not blue, it's brown, okay? They are located superficially. One more example of nests of melanocytes dermatoscopically seen as brown globules. Again, brown dots and globules can be seen also in melanoma. The difference is that they are atypical, irregular, meaning that we see dots and globules of different diameters, and also they are not arranged regularly. Why? Because in melanoma, the nests of malignant melanocytes are not homogeneous, are not equal in size and distribution as they are in nevi. Look at the histopathologic image on the left. All the nests of melanocytes are so regular, more or less the same size, and regularly distributed. In contrast, in the melanoma case, in the histopathology image, you can see a very large nest here, a much smaller nest here, very small nests just aside. So that's why the globules are atypical, irregular in melanoma. So brown globules correspond to nests of melanocytes immediately below the junction, diagnostic significance, nevi, and melanoma. Next one, streaks, peripheral streaks, corresponds to confluent junctional nest of pigmented melanocytes at the periphery of the lesion, indication of a radial growth. That's a very nice histopathologic correlation. Look here at the peripheral end of the lesion, the large nest, which will be then divided in two smaller nests, and this is how the tumor grows peripherally. So brown streaks and pseudopods, confluent melanocytic nests at the periphery. Let's move on. Brown structures again, but in this scenario we see these brown interconnected areas that consist of projections originating from a common base. These are the so-called leaf-like areas or spoke wheel areas that histopathologically correspond to interconnected nests of basaloid cells located where? Junction. Again, that's why they are brown, and that's why they correspond to superficial basal cell carcinoma. One more example of these peculiar structures and the histopathologic correspondence. Look at the interconnected basaloid nests. So leaf-like areas and spoke wheel areas correspond to interconnected pigmented basaloid nests at the junction, again, diagnostic significance, basal cell carcinoma. Let's move then to gray. Gray is another very important color. Again, we will see first some cases and then go to the explanation. This is a melanoma. The next image is something completely different. It's an inflammatory disease. It's a lichen planus. The next case are two examples of a regressive seborrheic keratosis, so lichenoid keratosis, lichen planus-like keratosis. The next one is again not neoplastic, it's a case of erythema dyschromicum perstans, and the next one is a fixed drug eruption. And one more case in which could be either regressed nevus or an LPLK or a regressed melanoma. So in all these cases that are very different in terms of pathophysiology and biology, malignant tumors, benign tumors, inflammatory conditions, there is again something in common. In this group of lesions, what is in common are these gray small dots or gray granules. And these granules are there in all these different entities because histopathologically there is the same thing, which is the so-called melanophages, macrophages that do a procedure which is called phagocytosis. They eat practically the melanin. And that's why in all these different entities we see precisely the same thing. And here are the histopathologic images. So it makes sense. Gray dots and granules correspond to melanophages and they can be seen in numerous tumors and entities. And finally, blue color. Blue corresponds to melanin deep in the dermis. That's why it's blue. Otherwise it would be brown or dark brown or gray. But deep in the dermis, blue. So now let's see here first the schematic. Here you can see large nests of cells that you don't know what they are. They are just pigmented cells. Deep in the dermis, large roundish nests. So what do you expect to see dermatoscopically corresponding to this schematic? You expect to see large globules of what color? Blue. So nests, okay? In the next schematic, you see a diffuse presence of melanocytes in the dermis, but here there are no nests. It's just diffuse presence of melanocytes. What do you expect to see dermatoscopically? Diffuse blue coloration, no nests, no network, nothing else, just blue color, structureless, okay? And that's the case. So this is the first correspondence, the first image we saw before. Nests, globules, blue globules, dermatoscopically corresponding to these large nests, and this is very significant diagnostically for basal cell carcinoma because this is the only tumor in which there are large pigmented nests in the deep dermis. In nevi, as I showed you before, the melanocytic nests are not in the deep dermis, they are in the superficial dermis. That's why the globules in nevi are brown, they are not blue. So blue globules, blue clots are a strong clue for basal cell carcinoma. And here is the histopathologic correspondence. One more example, blue globules correspond to pigmented nests deep in the dermis, basal cell carcinoma. In the other scenario, diffuse dendritic melanocytes in the dermis. Of course, this corresponds histopathologically to this very well-known blue nevus. Dermatoscopically, we don't expect to see blue globules. Dermatoscopically, we expect to see blue structureless, and this is, of course, the case in blue nevi. Diffuse melanocytes in the deep dermis. And finally, blue color of course can be seen in melanoma because melanoma cells obviously invade the deep dermis in an advanced stage, and therefore blue color might be seen also in melanoma. The difference is, as compared to blue nevus, is that we would very rarely expect the melanoma cells to infiltrate the dermis in a symmetric way. Usually the infiltrations will be much more dense in some areas, therefore intense blue color in some areas and less dense in other areas because the invasion of melanoma is not symmetric, obviously. So structureless blue corresponds to dermal melanocytic, dermal presence of melanin and might diagnostically be associated with blue nevus or melanoma, and blue nests correspond to pigmented basaloid nests in the lower dermis, diagnostically very strong feature for basal cell carcinoma. Okay, this is an overview of the main criteria we see in pigmented tumors, and this information in my view is very important as preliminary basic knowledge for other presentations that will follow and will focus on specific diagnosis. Thank you very, very much for your attention.